Hello and welcome. My name is Hilary Henley and I'm a global medical researcher with the Reinsurance Group of America. Today I'm delighted to be joined by Dr. Nathan Stitziel. Dr. Stitziel is Associate Professor of Medicine and Genetics at Washington University School of Medicine in St. Louis. His work focuses on applying next-generation genetic techniques to understand the inherited basis of cardiovascular disease. Dr. Stitziel is also a current research grant recipient of the Longer Life Foundation, a nearly 25-year collaboration between RGA and Washington University, funding cutting-edge research into the drivers of mortality as well as morbidity. Dr. Stitziel, thank you for taking the time today to talk to me about your research work. If I could first ask you, who or what sparked your interest in medical science? And then what inspired you to become involved in the field of human genetics and cardiovascular disease?
In terms of my interest in medical science, I think like most scientists, it really goes back to my childhood where I had an interest in trying to understand how the universe worked and how the world around us worked. I was one of those kids that was always taking things apart, trying to figure out how devices were built, and just really curious about the world around me. I carried that through into my studies and actually went to college as a math and physics major. So I was a double major in college, thinking I would go to graduate school and pursue a PhD in science. And as I got more into exploring different types of research, I really became drawn to trying to address research questions that were more relatable to humans and human health. And I found myself veering away from strictly a PhD in science and more toward a combined medical degree and a PhD to be able to perform biomedical research. In terms of how that relates to cardiovascular genetics, there is a connection there. So as I mentioned, I was a math and physics major, and I was really trying to find something where I could combine my interests in medicine and my interest in quantitative science. I was drawn to the cardiovascular system because of its relationships with physics. It's largely hemodynamics and electrical circuits, for example. And so I think intuitively I was drawn to cardiology. And then within cardiology, I was looking for, again, ways to apply data analytics to try and understand how fundamental things worked. And the field of human genetics was really exploding at the time that I was looking for, advanced research training. And I got into the field of cardiovascular genetics and been happy ever since.
Thank you. It's interesting to see how your background studies have drawn you to this area of medicine. Your most recent research work has focused on circulating levels of the protein SBP1 with multiple age-associated chronic diseases such as coronary artery disease, hypertension, diabetes, as well as dementia. How do you see these important discoveries influencing prevention and treatment of disease in the future?
Our work related to this protein, we call it SVEP, SVEP1, but I'll just say SVEP for short. Our work with SVEP really dates back about 7 or 8 years now, where we tried to identify causal factors or causal proteins for cardiovascular disease. Our lab uses a technique, or our lab uses an approach where we try and identify genetic factors that underlie human disease and then do translational studies to understand how those genes or proteins actually impact disease. And we make the argument that for a long time, we have been studying coronary disease or the surrogates of coronary disease in mice. And we've done genetic manipulations in animals to try and figure out what blocks the development of atherosclerosis in mice. And then many, many, many years later, we develop an agent against some target and we do a trial in humans. And oftentimes those trials are not successful. And the reason they're not successful is that humans and mice are very different. We diverged evolutionarily a long time ago. And the factors that drive human disease are not necessarily the same factors that drive disease in animals. And so our lab and others uses an approach where we say, well, if we want to study human disease, it turns out that humans are a pretty good model organism. So we try and understand the genetic drivers that underlie disease, and then go back to animals to try and figure out mechanism. So our work with SVEP began, again, about 7 or 8 years ago, where we initially discovered a genetic polymorphism or a genetic change in SVEP that related to coronary disease. We then knocked out the gene in an animal and could show that atherosclerosis comes down. And through a series of human genetic studies, have now really started to build a evidence base around SVEP1 and how it causally promotes disease in humans. And the more that we start, the more that the field has been able to study SVEP and measure SVEP, we are now appreciating that it is likely causally related to multiple chronic diseases of aging. And this is how our work was ultimately, in part, funded by the Longer Life Foundation because of its association with these multiple diseases of aging and longevity.
Based on your research work to date, realistically, how far away are we from incorporating biomarkers of age and longevity in humans in everyday medicine?
Yeah, it's a little hard to know how far away we are in terms of incorporating those biomarkers of age. A lot of work has to be done. But I think the important thing about SVEP is that it's not merely a biomarker. Our studies have shown that it's actually it's a causal factor that promotes these diseases of aging. And I think that's going to be important in the future because, we can measure biomarkers and there are lots of biomarkers out there that we can't impact. Or if we do impact those biomarkers and the biomarkers are not in a causal pathway, it doesn't matter. It might be a good biomarker for disease, but changing that biomarker won't actually move the needle on disease. So that's why we're so excited about SVEP, where we have shown that if we can reduce levels and or reduce its interaction with receptors that promote disease, we should be able to target those diseases.
From your own viewpoint, could you tell us a little bit about the impact of financial support from groups such as the Longer Life Foundation? How important is that support to the success of researchers like yourself, especially in driving pilot and feasibility research and then going on to secure those larger grants? Research
support from foundations like the Longer Life Foundation is absolutely critical at the beginning stages of these projects. The NIH and other federal funding agencies are very, I would say, risk averse. They don't like taking bets on ideas that don't have a solid basis of support. It's really hard to get very early stage projects funded by traditional funding agencies. And that's where foundations like the Longer Life Foundation provide such critical support to be able to develop a robust set of preliminary data to really support the ideas that you'll then pitch for larger funding. So again, it's really great that that these foundations like the Longer Life Foundation do support these cutting edge ideas.
And just, I suppose, to summarize, although it's difficult, in closing, could you describe your hopes and your aims in 5 words?
If I can get 6, maybe I'll say I can summarize it by saying we hope to leverage genetics to improve human health.
Dr. Stitziel, thank you again for taking time out of your busy day. We really appreciate it, and I hope all our viewers here have really enjoyed this presentation. Thank you very much.
Thanks, Hilary. It's been a lot of fun to talk.